Welcome to my presentation on microbes and miso. Miso is a common food used in Japanese culture, and people are most likely familiar with miso soup. Miso is formed from a special mold, koji. It is grown on most commonly rice but can be produced on barley or soybeans. For rice, the grains are cooked to a point where they are mushy and their starches are all released. The koji spores are then added and allowed to ferment. This whole process can take about 42 to 48 hours. At this point, soybeans that have been washed, steamed, and smashed are then combined with the koji, salt, water, and two organisms salt-tolerant yeast, Zygosaccharomyces roxy, and salt-tolerant lactic acid bacterium Tetragenococcus halophilus. This mixture is then added to containers in a warm and humid room where they are allowed to ferment and mature. Depending on the type of miso trying to be achieved, the fermentation can last as little as 1 to 4 weeks or as much as 6 to 12 months. The less timed fermenters produce a sweeter, lighter colored miso, while the longer time produces a saltier, darker colored miso. Tetragenococcus halophilus grows most effectively in environments of 5 to 10% salt but can tolerate up to 24%, which works great with miso since its salt contents fluctuate depending on which one is trying to be achieved. It is also a lactic acid forming bacteria, which helps with the degradation of unfavored sugars and promotes the desired flavors, and additionally encourages the growth of the yeast during the time of fermentation. Fermentation begins with catabolism of glucose via glycolysis. The NADH produced reduces pyruvate down to lactate, which helps suppress bacteria that would be detrimental to the growth of koji or yeast. The glucose from the rice or other media being used for the koji provide ample nutrients for the yeast to flourish. Alcohol is formed by the yeast, which enhances the miso flavor, and this is maintained in a low moisture environment to sustain the growth of the fungi. This leads to catabolic enzymes being produced, which affect the miso further in production. Rice that produces more amniotic enzymes due to the high sugar content and is used for sweeter miso, whereas koji made from soybeans will produce a higher quantity of proteolytic enzymes, which would be more suitable for savory flavored miso.
Hello, this is Juan, and this is microbes and foods for homework 2. So the food I chose was yogurt. So it is a dairy product. So some of the things it does is, provide nutrients, and it has benefits of protecting the immune system. One of them is it protects the good bacteria in the colon, so it doesn't remove it. And it consists of only two ingredients, that being milk and live culture. And we will be talking about those live cultures. So the first one is Lactobacillus bulgaris, I think that's what you call it, is the first of the vital ingredients in yogurt. It's gram-positive, anaerobic. It has the appearance of a rod that's long of filamentous. I think that's what I say it. And the second one is the Streptococcus thermophilus. Is the second vital ingredient in yogurt. It is gram-positive also. But this one is anaerobe. It has a bell's shortest shape. And lactic acid fermentation. This is the fermentation process to make yogurt. So what it does is it turns substrates such as the glucose into lactate or lactic acid. So an example of this would be yogurt. So those two previous phytobacteria, when put into milk, actually causes the two vital bacteria to produce lactic acid, and it will lower the pH of it and cause it to congeal. All right, and that's it for my homework assignment. Thank you very much.
Microbes and my food. Salami. Salami is a type of cured sausage typically made of coarsely ground beef and pork seasoned with spices like garlic, pepper, paprika, and salt. This mixture is stuffed into a natural casing made of hog or sheep intestines and cured for extended periods of time. Salami is believed to have its origins in Italy. These products date back to ancient Rome, and it has been made in various forms in Europe and North America. Curing salami takes time, usually ranging from weeks to months. During this process, the salami is hung in temperature-regulated rooms, low humidity environments, and simply ages as bacteria such as Staphylococcus aureus Staphylococcus psilosus and Staphylococcus carnosus, fully flavor the product through a fermentation process. Once the ideal point is reached, flour or mold is applied to the surface to stop photooxidation and prevent the meat from getting spoiled or toxic. I'm focusing on Staphylococcus carnosus. Staphylococcus carnosus is a gram-positive, non-sporing, facultative anaerobic bacteria that is part of the Staphylococcus family. It is generally found in processed meat, as it is a great starter culture in the production of traditional fermented sausages such as salami. It gives cured meats flavor and improves their chemical, physical, and microbiological stability. Staphylococcus carnosus can grow in various temperature ranges and extremely low concentrations of salt, which makes it a popular choice for meat processing factories. During fermentation of the meat, the Staphylococcus carnosus produces metabolites such as lactic acid, alcohol, carbon dioxide, acetate, fatty acids, esters, and sulfur compounds that give the meat its unique flavor, odor, and texture. Fermentation is a metabolic process that takes place in the absence of oxygen, which means microorganisms must use alternative pathways to break down food molecules. In the case of meat fermentation, as is in salami, the protein and organic materials in the meat are broken down by bacteria, producing lactic acid, carbon dioxide, and other organic acids, as well as ethanol and other alcohols. This mixture of products is known as the fermenta. The microorganisms consume the sugars, protein, and fat present in the meat. This happens through enzymes such as proteases, lipases, and amylases. And as the fermentation process continues on the meat, the lactic acid created by the bacteria provides a protective environment for other microorganisms and aids in avoiding the growth of toxic pathogens that could spoil the product. Genetic modifications have been used to improve the flavor, tenderize the meat, and improve nutritional content and shelf life of the salamis. These modifications can be done through bioengineering of the microorganisms, cloning, and genetic recombination.
We'll be discussing kombucha and acetic acid bacteria. Over the years, people have been constantly on the search for new, healthier products to improve their health and wellness. One of the numerous products that has been popular among consumers is kombucha, as it contains many nutrients, probiotics, and antioxidant properties. Kombucha is a fermented tea that is traditionally made with either black tea or green tea, fermented with a symbiotic culture of bacteria and yeast, which is called SCOBY, and sugar. A couple of the bacteria groups used for the fermentation process are acetic acid bacteria and lactic acid bacteria, both of which play a role in the taste of kombucha as well. Acetic acid bacteria are a group of gram-negative obligate aerobic bacteria. They're bacillus-shaped and produce acetic acid through oxidation of ethanol during oxidative fermentation. Acetic acid bacteria can be found in a variety of sources such as flowers, fruit, or fermented food. During the fermentation of kombucha, the symbiotic culture of the bacteria and yeast interact with each other along with the sucrose that's added to the tea broth, producing numerous acids, alcohols, esters, and polyphenols. The bacteria continues to ferment the kombucha even after it's ready for consumption, as the acid it produces helps to keep a low pH in the kombucha, which prevents spoilage and pathogenic contaminants. Aerobic fermentation is the metabolic process used to make kombucha. Sucrose is usually the choice of sugar used for the fermentation of kombucha and is the main carbon source. The sucrose is hydrolyzed by beta-fructofuranosidase and converts it into ethanol. Ethanol is then oxidized to acetic acid by the action of alcohol dehydrogenase and aldehyde dehydrogenase. When kombucha is flavored with fruits and juices, anaerobic fermentation occurs, producing carbonation. This carbonation helps to keep the alcohol content of the beverage within legal limits of a non-alcohol beverage.
Hello, class. So for my specific food item that I chose was cheese. Since cheese is a food that comes in many dishes, there are many types of different cheeses around the world. Cheese dates back about 5,000 years ago as remnants were found on people's teeth as tartar. Cheese is as amazing as it can taste. The process of cheese is even more astounding. Cheeses can be in two different ways. It's either made through the heating process or the unheated process, which is pasteurization, which is the heating one, and unpasteurized, which is the non-heating version, creating cheese. So some cheeses that are made from pasteurization would be mozzarella cheese, cheddar cheese, and then unpasteurized would be like feta cheese. Some ingredients of cheese would be the main one, obviously milk, But some other ones that would be kind of unexpected are salt and starch cultures. Starch cultures are basically like bacteria that start the initiation of the metabolism of milk to cheese. Um, that's why some people believe cheese is a living food item. The two main microorganisms are thermophilic and mesophilic species. Um, One of the thermophilic species would be Streptococcus thermophilus and Lactobacillus helveticus and Lactobacillus delbrueckii, which can withstand high heats like temperatures up to 132 degrees Fahrenheit, which would be the pasteurization process. Um, Streptococcus thermophilus and Lactobacillus helveticus are two or microorganisms which can produce cheese from lactic acid fermentation. The final products of these processes would be carbon dioxide, lactic, and acetic acid. And then once these processes occur, then this milk is then cooled down, which creates a gel-like substance on the top layer of the milk. So this would be in large containers when people would be creating milk. It would just be like a gel-like substance on the surface. And there are many combinations of bacteria, yeast, filamentous fungi, which create different cheeses. And each culture has their own unique way of creating cheese. Some genetic modifications that we've made over the time would be rennet. Rennet is used for the curdling process. In the 1800s, this was not an option as people used similar enzymes found in animal stomachs, which ultimately coagulated the milk similar to rennet. As research from scientists, we were able to create rennet using modified bacteria, and yeast cells. That's why I believe the process of cheese is amazing since it can be as unique as you want it to be, and every culture has their own way of creating delicious cheese. Thank you for listening.
Microbes in My Food, Kimchi by Ezra Kang. What is kimchi? Kimchi, a salted and fermented side dish, is a staple food in Korean culture. It originated in Korea thousands of years ago. Because of its longevity and shelf life, which extends to a couple of years, it has been a reliable food source. Since then, over 100 types of kimchi have been produced. Microorganisms within kimchi. The following listed below are all examples of lactic acid bacteria, or LAB. Lactic acid bacteria is a type of gram-positive bacteria that use carbohydrates as the only or main carbon source to produce lactic acid. Lactic acid bacteria are generally cocci rods and have a strong tolerance to low pH. Because the first step of making kimchi is a salt brine, all the other bacteria that cannot survive in these conditions are killed, so cross-contamination and spoiling is avoided. However, lactic acid bacteria can survive in these harsh conditions. How kimchi utilizes fermentation. Fermentation is an anaerobic process that allows glycolysis to continue without oxygen. Fermentation is less efficient than the aerobic process of producing ATP, but it is also very helpful due to its lack of need for oxygen. The natural lactic acid bacteria in the vegetable used to make kimchi, such as radishes and cabbages, process the carbohydrates present in the ingredients used to make kimchi. The lactic acid produced is what gives kimchi its unique taste. In the picture below, you can see carbonation. With proper storage, kimchi can last over a year in the fridge. Thank you.
An extremely ubiquitous staple in many people's diets, primarily produced in Brazil, Ethiopia, and Indonesia. It's primarily produced in Ethiopia using a method known as garden coffee, wherein coffee is grown by small communities of farmers near their residences. It's a multi-step process before coffee is ready for consumption, involving drying, fermenting, and roasting. The fermentation step serves to break down the skin of the coffee bean, and this can be accomplished by numerous different types of bacteria and fungi. One specific genus involved is the lactobacillus bacteria, and this breakdown of the skin is known to improve the coffee's flavor, diversity, and complexity. Lactobacillus plantarum is a commonly found bacteria in many food products, as well as a member of the human microflora. It's aerotolerant, about 6 micrometers long, bacilli, which tends to occur in small chains. It's commonly used as a fermentation starter, and it's the starter colonies are put into a water bath with the coffee beans, and the skin of the bean serves as a substrate for the bacteria to grow upon. This is a delicate balance between under and over fermentation. Being a lactic acid bacteria, L. plantarum's primary metabolic process is the breakdown of carbohydrates into lactic acid. It uses a pathway called heterofermentation to accomplish this, and the most basic pathway involves the breakdown of simple glucose into lactic acid. This follows a series of steps, and ATP is produced in this process, giving L. plantarum the energy it needs to survive. This makes an aerotolerant anaerobe, and the production of lactic acid is correlated with high concentrations in fermented coffees.
Hi everybody, this is about how vinegar gets its tang. We're going to talk about wine vinegar in particular, and the star of the story is Acetobacter aceti, a rod-shaped bacteria discovered by Louis Pasteur in 1864. It's gram-negative, paratrichous with its flagella, and it likes to work in a pH range of 5.4 to 6.3. It converts ethanol to acetic acid. Okay, so here's what happens. We start with wine, all right? And then we add wine into a flask with yeast. The yeast converts the glucose in the wine or fructose in the wine to a 15% alcohol solution. Then we add a mother, which is a living sponge-like piece of colony of Acetobacter aceti, and that does the conversion. When that's done, we have acetic acid, and that means we've got vinegar. Let's look at the chemistry. Again, we start with ethanol, which is 2 carbons with a hydroxyl group on the end. And then a water molecule takes away one of the hydrogens and leaves us with an aldehyde double bond, and that's ethanal. Okay? The next step, the final step, and this is done with the help of enzymes in the Acetobacter, is the double bond gets a hydroxyl added onto that carbon. Now we have a carboxylic acid group. That's acetic acid. That's what makes your vinegar so delicious.
Most, if not all, foods we consume have a long history of a relationship with microbes. Blue cheese, like Gorgonzola, Stilton, or Roquefort, specifically wouldn't exist without fungus. Roquefort, a type of blue cheese developed in France as early as the 10th century, is made by combining sheep's milk, rennet, salt, and the fungus Penicillium roqueforti. To produce Roquefort, typically all the ingredients are added and the cheese is left to sit and curdle. The whey is drained, then the cheese is poked a number of times to allow oxygen in for the fungus to grow. It is then left to sit as it is aged for months. Once the cheese has finished aging, the characteristic blue-green veining is produced by the growth of the fungus P. roqueforti. P. roqueforti is a saphotrophic fungus that uses chemoheterotrophic extracellular digestion to obtain nutrients. This means that it produces acid, which it excretes out into the environment to break down the organic compounds in the milk and curds. P. roqueforti is aerobic, thrives in wet environments at a medium-low temperature with a neutral pH. The fungus is initially microscopic, but as it obtains more nutrients, it starts branching by hyphae, which are long filamentous structures that appear velatinous in texture and blue-green in color. Once the cheese is fully aged, it is typically consumed raw with the live fungus and all of its products. The byproducts for the breakdown of the proteins, fats, and carbs in the cheese by the fungus cause the cheese to have the distinct and pungent taste and aroma. Other byproducts include small amounts of mycotoxins and even some byproducts like mycophenolic acid, which has been isolated to have possible medical uses. I'm going to tell you about sake production today. Sake is a fermented Japanese beverage, and it's made from rice, water, koji, yeast, and of course the brewers, who are a very important part of the process. First, the rice actually is harvested. Then it is transported to a brewery where the rest of the process is overseen by a master brewer known as a toji, who is assisted by a skilled force of brewers. Sake actually is a process that needs a lot of close attention and care at all hours of the day and night. So skilled brewers are a very important part of this process. Once the rice makes it to the brewery, it is polished to remove some of the outer part of the rice. It's then soaked, washed, and steamed. And then koji is scattered onto the rice. And we'll talk about koji in just a moment here. Then yeast is added and multiple parallel fermentation happens. Sake is actually the only alcoholic beverage that does both saccharification and fermentation at the same time. After this happens, the second fermentation happens in vats for 3 to 5 weeks, and depending on the type of sake, it may be filtered or pasteurized. It remains in the tank for 6 months after that, and water is added to get it to a proper alcohol content, and then it's bottled and distributed. So Aspergillus oryzae is known as the national fungus of Japan. It is what makes the koji mold, which is so important for sake brewing. It's a filamentous branching fungus, an obligate aerobe, and it's also used in other fermented products like soy sauce and miso. In the old days, it was inoculated passively from the air or from the rice, but today it's usually gotten from manufacturers. So although saccharification and fermentation both happen in sake production, I want to talk about saccharification. So in this process, starch needs to be broken down to be fermented because it is just needs to be broken down into simple sugars in order for that process to happen. So substrates include the starch from the rice as well as amylase, which is an enzyme that's produced by the koji. So the koji produces this amylase, and then the products of this reaction are going to be glucose and other simple sugars. This prepares the sake for its second fermentation. Thank you so much, and kanpai.
I wrote my paper on bacteria used to produce yogurt, and I decided I would like to give it a try for myself. So this PowerPoint presentation will just kind of go over that process. A quick note, my family is vegan, so I did use a soy milk to make my yogurt with, rather than dairy milk. So just 2 ingredients, 1 quart of soy milk and 4 probiotic capsules, the kind that, people just take as probiotics. And you'll see a little bit about that. The recipe is here. Basically, you just mix the milk with the contents of the probiotic capsules and then pour that mixture into jars, place the jars into the Instapot, and then cook in the yogurt setting on the Instapot for about 8 to 12 hours. So here are just some pictures. Here is the soy milk that I used. Here is the probiotic that I used. And you can't see this label very well, but it lists all the different bacteria that are in this particular probiotic. For a product to be labeled yogurt, it does need to contain 2 bacteria by law, one of which is the Lactobacillus bulgaricus, and then there is another one that by law must be in a product for it to be legally labeled yogurt. And this probiotic did have the Lactobacillus bulgaricus. I'm not sure if it had the other bacteria in it, but there were about 10 different types of bacteria in the probiotic. So here's just a picture of the probiotic capsule unopened. So there's a gelatin, that keeps the bacteria in it. So to use it in the recipe, I opened the capsule. I don't know if you can see that here, and here are the contents. So the contents are what was mixed with the milk, not the gelatin capsule. And then here is a picture of my, what went into the Instapot. It's just the milk mixed with the contents of the probiotic capsule. And then I did set the Instapot for 12 hours. So it should, the process takes 8 to 12 hours. So I just did it for 12 hours. And then here is my product right here. And I tasted it, and since the only ingredient was soy milk and the probiotic, it really didn't have a lot of flavor, which was fine. I mean, it did taste good, but then I doctored it up with some maple syrup and some blueberries and had a great breakfast.
This is a presentation on the microbes in my food. Manufacturing companies are using microorganisms to make a range of food products. This process is known as fermentation. Cheese is made from milk that has acid added to it and goes through multiple processes to become cheese. All cheeses start with milk, but it is the bacterial cultures and the amount of acid that is added that is different for each type of cheese. Fermentation of lactose to lactic acid by lactic acid bacteria is an essential primary reaction in the manufacturing of all cheese varieties. There are many microbes that could be found in cheese. Lactic acid bacteria are a kind of microorganism that can ferment carbohydrates to produce lactic acid and are currently widely used in the fermented food industry. Lactic acid bacteria are used as starter cultures in the process of making cheese. Milk is coagulation by heating or by the formation of adding acid. This helps the coagulation process happen faster and it becomes firmer, which results it to become a curd. These cheese curds are then added to molds where the liquid can be drained. These molds are also what gives the cheese its shape. There are some cheeses that can go right on the grocery store shelf to be sold, and there are some cheeses that need to be aged for months, and even some aged for decades. Lactic acid is an aerotolerant anaerobe which plays an important role in the process of cheese making and the fermentation process. Through fermentation, the pH of milk is lowered and becomes more acidic. The longer cheese ages, the less lactose is left over because it's been used by the bacteria. By the time an aged cheddar or Parmesan is ready to eat, hardly any lactose remains in the cheese.
Microorganisms have been used in food production to bring us various types of foods such as cheese, bread, and kimchi. Some foods even contain live microorganisms in them such as Yakult. To make Yakult, only a few ingredients are needed, but the most important one being Lactocase bacillus paracase strain Shirota. The bacteria is fermented for 6 to 9 days, and one bottle contains at least 20 billion L. paracase strain Shirota. So first, milk arrives from farms and the process for making Yakult begins. Lots of machines and workers are involved in the process until eventually it is bottled, packed, stored in a cold room, and comes to people in a few different ways. The first way is sending it directly to supermarkets or convenience stores where people can buy them. And the second way is very unique to the company where there are Yakult ladies that deliver Yakult to people's homes, which would build a loyal customer base. L. paracase is a rod-shaped, gram-positive, non-motile, non-sporing bacteria that can grow under anaerobic and aerobic conditions. It can be found naturally in the GI tract of humans and animals. By the time Yakult is ready to be drank, there are still live L. paracase bacteria found in the bottle, which would help suppress bad bacteria, increase good bacteria, and also aids in digestion. The metabolic process Yakult relies on is lactic acid fermentation. So skim milk is first sterilized, and then the starter culture L. paracase strain Shirota would be added into that milk. The strain is a lactic acid bacteria and produces lactic acid through fermentation, which would cause the milk to taste sour before other sugar and flavorings are added into the mixture. First, lactose from the milk will be broken down to glucose, and from there, glycolysis occurs, resulting in two pyruvates. Pyruvic acid is reduced to the final product lactic acid by NADH.
Hi, my name is Maham, and I'll talk about microbes and food. So, food is very important for us because it gives us nutrition that allows our cells to produce energy for us to do basic tasks. Of course, we know moldy food is bad to consume. There are microbes that facilitate that smell and mold we often see on food. While we have an increase in population, there has been an emphasis on food production and food preservation. In my presentation, instead of food process, I will focus on food spoilage, but in particular meat spoilage. Meat often comes from CAFOs, which are essentially industrial livestock for human consumption. Animals naturally carry salmonella and E. coli in their intestines. Meat is one of the most perishable food because of its condition that attracts microbes. Food spoilage is caused by microbial growth or by actions of microbial enzymes in food. The presence of slime and colonies are caused by degradation of macromolecules by microbes. Gastroenteritis is common with these pathogenic microbes when they are ingested. This causes nausea and diarrhea. Meat has the ability to obtain high water content and nutrients on its surface, which is ideal for microbial growth. There are many kinds of food-spoiling bacteria, but the most common one is bacterial species Brochorothrix. Brochorothrix thermospecta is a common spoilage bacteria that are in meat stored at cool temperatures. B. thermospecta is a non-sporing, non-motile, gram-positive, and facultative anaerobe. Being facultative anaerobe means that the bacteria can grow in conditions that do or don't have oxygen. There's a metabolic switch in B. thermospecta when atmospheric conditions switch from dominantly anaerobic to aerobic metabolism. Anaerobic metabolism will release lactic acid, and aerobic metabolism will release acetyl diacetyl. These both give spoilage meat its strong odor. Recent studies have shown that biotechnology has been used to use bacterial cells and biosensors that detect and respond to spoiled meat. Understanding that microbes are everywhere and how they metabolize are important so we can place appropriate measures to keep food and meat in a safe condition and to reduce food spoilage. Thank you.
There are many different brands of kombucha. Kombucha is a fermented drink that has a lot of probiotic bacillus, coagulans, and bacteria that are capable of fermentations, such as Lactobacillus naegeli and Gluconobacter. The fermentation process of kombucha starts when symbiotic culture of bacteria and yeast are added to sugary tea. Bacteria reported in kombucha cultures include acetic acid bacteria and lactic acid bacteria. Kombucha uses green, black, and white tea as their base for fermentation. The different components of yeast and bacteria, tea base, manufacturing process, and the probiotic addition create the potential for a wide diversity of bacterial and fungal compositions. Tea polyphenol levels and metabolite contents. Lactobacillus is part of the lactic acid bacteria group. These bacteria are comprised of gram-positive, catalase negative bacteria that produce lactic acid as the major metabolic and product of carbohydrate fermentation. They are gram-positive, non-spore-forming rods and Coccobacilli with a G and C content of DNA usually below 50. Catabolism is at its roots the breakdown of complex molecules. It is the breakdown of complex substances to more simple parts such as glucose, amino acids, and fatty acids. And these substances being broken down into their simpler forms help create substrates for metabolic pathways.
Microbes and food. Chocolate production begins with the growth of cacao beans in tropical areas requiring high rainfall, high humidity, and warm temperatures. The production of chocolate is then split into 5 processes: fermentation, drying, roasting, grinding and mixing, and then conching or tempering. Fermentation is the most biologically relevant, being split into 3 separate phases, all reliant on a different type of microbe. Production of acetic acid from phase 3 is crucial to chocolate production as it decreases the pH in the cacao beans and activates the pH-dependent enzymes that produce the flavor and aroma precursors. Acetobacteriaceae is the primary family of acetic acid bacteria, with the most abundant species present in chocolate fermentation coming from the Acetobacter genus. Unlike other acetic acid bacteria, Acetobacter species are able to oxidize the ethanol in high acidity environments. A majority of the acetic acid bacteria, along with the other microbes present, will be killed off during the roasting process. Ethanol oxidation occurs in 2 primary steps, with 2 additional steps that reset the important enzymes and molecules. These first 2 steps are thought to happen within a complex, as the acetaldehyde produced by step 1 is not released into the environment, only the acetic acid produced by step 2, which can then either just go into the environment or be further utilized as acetyl-CoA in the citric acid cycle, and that is the use of acetic acid bacteria in the production of chocolate.
Hello everybody, today we'll be talking about the role fungus plays in food production. Specifically, we will be looking at baker's yeast. Humans have been making bread using yeast for thousands of years. Without utilizing the fermentation process, bread won't have its rise and shape. The main ingredients of flour, water, yeast, and maybe some added sugar and salt provides an incredibly important staple for human beings. Introducing yeast in dough yields a bread product that is lighter, airier, and more tasteful. Your baker's yeast can typically be found in your local grocery store, where they lie dormant on the shelf in a jar or packet and wait to be hydrated again. Yeast cells digest food to obtain energy for growth, and they are so successful in baking because they love sugar. The capability to ferment sugars into ethanol is a key metabolic trait of yeast. The yeast cells are a critical component in the fermentation process that converts sugar to alcohol and carbon dioxide. The carbon dioxide gets trapped in the sticky unbaked dough, which forms tiny little air bubbles in the wet dough, hence the rising dough. Baker's yeast, specifically Saccharomyces cerevisiae, can be found in abundance in nature before being introduced to bread dough. S. cerevisiae provides a variety of enzymes that enable carbohydrates to be broken down, producing sufficient carbon dioxide to give bread its characteristic texture. Metabolically speaking, S. cerevisiae is a facultative anaerobe and undergoes aerobic fermentation in the presence of oxygen and sugars. Once consumed, some strands of S. cerevisiae are also relevant probiotics, which are not only safe but also provide health benefits by restoring the gut microbiota by positively shaping the gut microbiota. We know that the process of fermentation begins by the fungus activating enzymes that will convert sugar into carbon dioxide and ethanol. So these 2 pictures kind of summarize the process by which during ethanol fermentation, yeast cells complete glycolysis to produce 2 ATP molecules, an NADH and pyruvate. The pyruvate is then converted to acetaldehyde, which is then reduced to ethanol, and in the process oxidizes NADH back to NAD+, which can then be used again in glycolysis to make more ATP.
Hi, my name is Dawson. This is my Microbes in My Foods project, and I'll be talking about pickled radishes, lactobacilli, and fermentation. Radishes have been around for a while, originating from China and spreading across Europe. Coming in many different shapes, sizes, and colors, radishes have been a staple for ingredients in different foods. In the 17th century was when a side dish was taken by storm across Asia, the pickled radish. This fermented vegetable was a delicious side dish that had a unique taste, texture, and overall health benefit when consumed. The microbiology behind the pickled radish is during the process of fermentation. A tiny microorganism called lactobacilli is the reason why we are able to eat pickled radishes. They are found in milk, foods containing milk, and fermented vegetables. They belong to the Lactobacilliae family and is a rod-shaped gram-positive aerotolerant anaerobe bacteria. They produce lactic acid, which helps the microbiota of the human gut. Fermentation is an anaerobic process that allows glycolysis to continue without oxygen, creating lactic acid in the process. So when radishes are being fermented, the lactobacilli is breaking down the natural sugars, and that is why fermented fruits have such a high concentration of lactic acid.
Hello, microbiology. Today I'm going to talk to you about kombucha because it's one of my favorite beverages and it's becoming super popular in the mainstream. You find it in pretty much every store and people are also brewing it at home. Without microorganisms, though, we cannot have kombucha because kombucha is the fermentation of tea, and fermentation is the process where microorganisms break down and digest substances. So kombucha is really cool because there's a two-step fermentation process. So the first step is where the yeasts will break down sugars into ethanol, and then the second step is where the bacteria take the ethanol that the yeast produce and produce acetic acid and lactic acid in the process of lactic acid fermentation and ethanol metabolism. There's a byproduct of cellulose from this, and it creates a thing called a SCOBY. And a SCOBY is really cool because it's made of cellulose, and it's a media for the bacteria and the yeast to thrive on inside the kombucha. So there's one specific type of bacteria that I really want to talk about today, and it's the acetic acid bacteria, known as AAB. It's interesting because it plays a role in the second fermentation of kombucha, that's where the acetic acid is formed from the ethanol that the yeast previously had formed. A lot of winemakers actually don't like this type of bacteria because it's known to produce a vinegary taste, which we like in kombucha, but we don't really like in our wines and things like that. So acetic acid is produced by two catalytic reactions that involve pretty much the breakdown of ethanol into acetic acid. You also are going to be first oxidizing the ethanol with a protein ADH to acetaldehyde, and then after that you oxidize the acetaldehyde again into acetate, and then acetate is going to be the base for the acetic acid production.
Hi there, my name's Hillary, and this is my report on microbes in beer. I selected this topic due to my personal connection to beer brewing. Coming from a family of hop growers, I had the opportunity to observe my dad's attempts at brewing beer during my childhood. Not always were they that great, but he tried. This has piqued my interest in exploring the role of microorganisms, particularly yeast, in the conversion of wort into beer. The focus of this research will be on the metabolic process of yeast species, including Saccharomyces cerevisiae and Saccharomyces pasteurinus. Brewing beer is a multi-step process that involves boiling malted barley and hops to create a sweet liquid known as wort. This wort is then cooled and yeast is added, which consumes the sugars in the wort and produces alcohol and carbon dioxide through the process of alcoholic fermentation. The use of different yeast strains in the brewing process can greatly impact the flavor and aroma of the beer. Yeast strains can produce a spectrum of flavors and aromas, including fruity, spicy, crisp, or neutral. And the conditions during fermentation, such as temperature, also plays a role. Certain beer styles may incorporate bacteria like lactobacillus and pediococcus, which contribute to the sour or acidic characters of the beer. The brewing process is influenced by various types of microbial interactions, including associations, symbiotes, and diamibiotes. These interactions have a substantial effect on the final quality and characteristics of beer, as demonstrated by the positive impact of symbiotic relationships between yeast and bacteria on the beer's flavor profile and stability. Conversely, disymbiotic relationships between yeast and bacteria may lead to spoilage and decline in the beer's flavor and aroma. Thus, a comprehensive understanding of these microbial interactions is essential to guarantee the consistent production of high-quality beer. One specific organism that plays a critical role in beer brewing, is the yeast species, Saccharomyces cerevisiae. This yeast species is commonly used for the alcoholic fermentation of wort, producing beer with a well-balanced flavor and aroma. Furthermore, S. cerevisiae has a unique ability to consume a wide range of sugars, which results in a high degree of ethanol production, making it an ideal yeast for beer production. Additionally, S. cerevisiae is known to produce several flavorful compounds during fermentation, including esters, which contribute to the fruity characteristics of beer, and fusel alcohols, which provide the beer with a warming sensation. In terms of biology, S. cerevisiae is a spherical or oval-shaped yeast cell that reproduces by budding. It has a single circular chromosome and a well-defined cytoplasm membrane. S. cerevisiae can adapt to different environments, including high sugar environments, and it has the ability to grow in the presence of high levels of ethanol. In conclusion, S. cerevisiae is a versatile and important yeast species that plays a significant role in the food and beverage industry, as well as in science research. Its ability to ferment sugars and produce carbon dioxide and ethanol makes it a valuable tool for various applications, including biofuel production. To wrap up, beer brewing is a complex process that is greatly influenced by the metabolic process of yeast and other microorganisms. Saccharomyces cerevisiae is a critical yeast species in the production of beer, producing ethanol and a range of flavorful compounds during alcoholic fermentation. Hops, being one of the key ingredients, have a profound impact on the flavor, aroma, and bitterness of beer, making them a beloved ingredient in beer brewing. I never appreciated how cool it was to be able to see how hops are harvested and sold to beer producers until I was old enough to understand the process. Thank you.